Yeah, thank you so much for bringing me here today. And so I'm going to talk a little bit about like why we are building software infrastructure to support spatial technology in biology, and that really goes deep into why we developed Shelter Suite. As a little bit of introductory background, I think whenever the field adopts a new technology, and for in this case, spatial omics, there is a lab, there are a few labs that develop a technology, spatial transcriptomics, and that goes into industry, and the industry adapts that and builds a lot of cool tools that are then all of a sudden accessible to so many other labs. And so as biologists or clinicians, we think about what are the samples we have, what can we do with kind of this type of technology, and that goes into data analysis and all sort of like difficult and easy ways of understanding like the spatial data. I think there is a layer in between that is often forgotten, and that is kind of the whole software engineering that makes it possible to go from the technology point of view and generating the data into like using and reusing data analysis strategies and methods. And so our focus right now is a little bit more on this software engineering point of view for spatial omics, which I think is essential if we want to like make a very strong foundation for data analysis, spatial data analysis. All right. So why exactly focus on software engineering? I think this is the time to do it. And there are definitely a few reasons to do this. So I think there are many different spatial technologies. It's very different compared to, for example, the single cell field where almost everything converged into one or two technologies. But in spatial, there are so many different technologies and they're very complementary to each other. So it's not that one is so much better than the other one. It's more about which one do you need for your specific questions. So they have difference in the resolutions, output, throughput, and here you can kind of like see how many of them have been developed. And actually, this is really outdated because there are so many more that have been developed only in the last couple of years. The fact is also that the number of data sets and the methods as a consequence have really exponentially increased. And so it's really now or never if we think about what is the kind of like the unifying engineering solutions we need. So if we look at the number of data sets and the number of kind of like spatial methods or analysis types that have been developed, it's really been exponentially increasing in the last years, and it's keep going. It's going faster and faster than ever. And I think last but not least is that spatial omics data is really complex. It's a lot more complex than I think single cell RNA-seq, and for a number of reasons. And just to show you why, a lot of people show this thing where you have the bulk single cell and spatial, and the resolution of this one is really bad because I think the figure is really bad. I think this analogy is really bad. It really oversimplifies, if you ask me, the difficulty of understanding spatial omics data. And so what I would like to do is give you another analogy, although maybe we shouldn't make any analogy. But I want to start with kind of like using like just some old bricks, and that is for me is kind of like RNA-seq. And RNA-seq can be really powerful. If you think about old bricks and you think about the ruins of what we learned about the Greek and the Roman empires, there's actually quite a lot you can learn simply from looking at like some kind of bricks. But if you really want to go more in detail and understand like what is it that makes these bricks, or what is it that makes these buildings, and what you can do with it, you can go to single cell RNA-seq, really pick apart all the different parts. You have the bricks, the cement, the concrete, whatever you want. You have a very clear view about what is needed to build something. But it's functionally, it has some clear limitations. And I think it's only when you put it back together in kind of like a structure that you see that you can build actual houses. These are the functional units that are made from these like individual parts. But also these functional units, in this case, houses, can be very different, in shape, can be very different in purpose. But that's only part of the picture. I think the larger picture is you need to put these houses back in their original location. And they can be in the suburbs, can be in the city. The city itself can also have different shapes, can have different ground plans, can have different bottlenecks, like traffic jams, whatever else. And here, for example, we have Rome, we have Boston, kind of like similar-ish in kind of like in size maybe, but very different in terms of what they are designed for and what they do. And then this is all analogies with all the spatial omics. Is you can create this pretty pictures and look at it and say like, wow, that's great, but it doesn't really give you a lot of insight. And so that's this other additional layer on top of it. You need to find ways to annotate your spatial data to get meaning out of spatial data. And you can do this in one way. You can annotate it and you can identify the byte paths. In other ways, you can really make more like abstract representations of your spatial data. And that's what I'm showing here. And this is where you really get meaning out of spatial data. So all of this, I think, is probably a better way of understanding all the complexities that goes into representing and understanding and using spatial data for biological insights. All right, and so does this apply, this complexity, does it apply to spatial data sets? And I'm calling this data sets of the future, but it's not really the future. We are already making like very complex data sets. So I think a spatial data set by itself, because of its nature, is already a little bit more complex than single-cell RNA-seq, as I said before. But nowadays, we can make these very complex data sets from serial sections of the same tissue. You can make multiple sections. And you can then, for example, create a single-cell RNA-seq data set. You can use one for a Visium type of approach. So this is more like a unbiased type of spatial transcriptomic analysis, or a more like with the new Xenium platform, you can go into a more targeted approach. There are definitely a lot of other examples. Here is another example from a Belgian group where they combine spatial transcriptomics with mass spec. And as a last one, I'm just showing this one, which I think is pretty cool, where you combine MERFISH with electron microscopy. So it's all combining like these different modalities with each other and thinking about what can we learn from these multimodal viewpoints. This is one of the only slides that I'm showing about the actual work in the lab. Besides this software engineering, is really we think about, and I think also like Nils kind of like introduces a little bit, what can we learn in the cancer field? And just to give you an idea, we work with triple-negative breast cancer together with Boston Medical Center. And so these are two research biopsies. So one research biopsy, and this is what people have been doing a lot, we can turn that into a single-cell RNA-seq data set. That's pretty cool by itself, because these are core needle biopsies, very small. But the moment you turn it into a single-cell RNA-seq data set, it's gone. And if you're lucky, you have like 4,000 or 5,000 cells with like 10,000 genes. So it's not bad. It's not great either, as I will highlight now, is that we can use one of these other core needle biopsies, kind of straighten it up, put it into a mold, and serially section through the whole triple-negative breast cancer biopsies, where you can get hundreds of individual slices through them. What we did for this specific project is we took 8 of these sections, kind of like throughout the whole C-stack, with like 80 or 90 micron in between each C-stack. And so one individual slice that you see here captures about 40,000 cells, or can easily capture about 40,000 cells. And that's actually less than 1% of the total biopsy. So this is one of the reasons why we made this 3D representation. So we can turn this into a 3D spatial data set. And then what you can also do on top of that is, as I said, we have multiple additional slices in between. So you can turn these additional slices into another Visium if you would like to. You can add an immunofluorescence stain. You can do imaging mass cytometry. So that's a spatial multiplexing protein type of analysis. You can ask your pathologist, can you please look at one of these slides because we identified something that's interesting. Can you see what you think about it? This is just all to say that I think it's really powerful that we can make these data sets, and that it's really amazing where we can go from a single cell RNA-seq data set where we have about 5,000 cells, where we can go all the way to a 3D digital biopsies with multimodal information. So it's like 1,000 times more informative simply from a quantitative point of view. All right, so this all brings me to the fact, showing this, like we early on started thinking about how can we build a unified and standardized data structure to represent and combine all these different technologies and data sets in a really efficient manner, and something that we can use in our daily lab life. So maybe some of you are like very confused, like what exactly do you mean? And I understand that. So I'm going to go a little bit more deeper. And this is where I'm basically saying, like, we made Giotto Suite, which is a multi-scale and technology agnostic spatial multi-omics analysis framework. So it consists of a number of kind of like a modularized way R packages that together form your Giotto Suite, and that can do all sort of things. But as a 1 or 2 slides background, we created this as a way on top of Giotto, our original toolbox. So it keeps that very comprehensive analysis framework where you can do all sort of like clustering, identification of spatial neighborhoods, cell-cell communication, everything that was previously encoded into Giotto. We have about 1,000 new users in the last month alone. So a lot of people are just like getting onto this and are liking what we're doing. But also, like, it was one of the first spatial frameworks It was the first release was actually before you saw this massive increase. And at that point, there were only a few data sets. And I think I analyzed all of them. There were like 7 or 8. And I thought I had it all figured out. It's like, oh, this is super easy. We don't need to have anything else. And now I'm going to get back to some of the software engineering approaches. So originally, it was simply like you generate data. I had an R package. You could just feed it in. And it was like, oh, that's super easy. I think nowadays we're splitting up Giotto in the kind of like the data processing end in this modular processing analysis where you can do separate things with separate packages and you can use all of them or some of them. But perhaps more importantly, and that's what I'm talking about most of the time today, is that we really needed different ways to represent the underlying data so that we have like independent and interoperable data structure for the different data types that we were working with to make it as flexible as possible for all the different, and I showed you some of these future data sets to work with them in a very efficient way. Last but not least, we also work on kind of like the data storage and think about how we can do this in a very flexible and scalable way. And so altogether, Giotto Suite kind of like represents its data generation, data storage, data representation, data processing in one way or the other. All right. So going into the actual figures and data for Giotto Suite is, so I'm going to show you a couple of things about how exactly do we represent data and how do we work with this spatial multimodal data. So in the way we represent the data, I'm going to show you like how it allows us to work with multimodal data, how we can work with multi-scale data, the hierarchical nature of biology itself, and how we can easily use it to do registrations and segmentations. I'm also going to highlight how Giotto tries to be as interoperable as possible, showing some of the interactivity tools that we have and how we are making this as scalable and flexible in the future. All right, so going to the data representation, this is probably the core framework of Giotto Suite as at least one of the modules. All right, so as I said before, the technology in biology and in the spatial field is really at multiple scales, right? The technology can be almost a micron all the way down to a few nanometers if you think about individual spots of transcript for some of these technologies. At the same time, biology also happens at multiple scales where you have larger macrostructures all the way down to suborganelles, and these suborganelles can be further like subdivided. All these different technologies that I showed before can have very different ways of data types, and the way you represent them is one thing where we thought about what kind of data structure do we need to efficiently represent images, points, intensity values, and shapes. And all of that together, you can kind of like stack it in a vertical way to create this vertically integrated connectome from all these different genetic layers, epigenetic layers, and so on. All right, so what is the core principle? So one, we don't actually aggregate any type of data. We keep the feature space and the spatial unit space completely separate, where the feature space can be any type of data. This can be your image. This can be also your points. This can be your intensity values, depending on the spatial technology that you use. And spatial can be anything that you define yourself and can also be multiple. Oftentimes, people like to go to a single cell, but it can also be your nucleus. It can be a larger domain. It can be some kind of grid, or it can be a combination of all of them. And I'm going to show you some of these examples later on. And so it's, as I showed you before, like the spatial units are very flexible. So it can be cell segmentations, can be spatial areas, spatial binning, but it can also be a combination thereof. And the points and the features, for example, are points, intensities, and so on. And then you can start aggregating it however many times you want in whatever type of combination you want, and use that type of aggregate analysis for your typical more downstream analysis or integrative analysis. All right, so I think conceptually thinking about how this is supposed to work is one thing, and I think we and others have done this, but really showing how it works and showing that it can work is actually another complexity. So I'm going to hopefully convince you that we have a number of examples to show you how this framework can work and how people can build from this in the future. All right, so here's a kind of like a simple first multi-scale analysis. using merfish data, where merfish data, you have this individual transcripts, and then you can assign these transcripts to individual cells. But these individual cells can also be individual nuclei. It can be individual, like larger domains. It all depends like how you segment and what type of additional annotations you have on top of everything. So we did this for the same kind of like subset at merfish data here, where you have nuclei, you have cells, you have a niche, you have a domain. So you might identify this specific niche as something that you like, is interesting. And you can very easily, using these Sankey plots, link their specific associations between all these different spatial annotations. So that's on the cell level or like higher up, but then you can also think about what if I'm interested in looking at the cell and nuclei. And here we do another very simple analysis, just showing the framework again. That what we can do is, for example, look at the individual transcripts and where are they enriched? What are the transcripts that are enriched in the nucleus versus the ones that are more enriched in the cytoplasm? And that can be in any type of, in your whole tissue or in a specific domain, as I showed you before, if you want to query for that one, or in some kind of other niche, or based on pathology information. And in this specific example, what we saw is that the transcripts that are like enriched within the cytoplasm are associated with the endoplasmatic reticulum, while in this specific data set, it was cell death, regulation of cell death. These transcripts were enriched in the nucleus, which might make sense since we're looking at a merfish breast cancer data set. So maybe there is some ongoing active apoptosis or some stress-related induced factors that are happening at the biological scale. Another kind of like simple thing that you can do is what I like to call just spatial in silico kind of analysis of single cell versus single nucleus analysis, where you can say, I want, as a spatial unit, I want a whole cell. Let me see what kind of like annotations you get. But if you do something similar, but you only take the nucleus, do you see the same cell types or not? And it's a little bit surprising, or maybe not for some of you working in this field, is that there's definitely not a one-to-one, using the same kind of like K, same kind of number. There's definitely not a one-to-one correspondence between the cell state and the nuclear state, indicating that where the cell is going versus where the cell is right now might be two different concepts, which I think is kind of interesting and kind of maybe alludes a little bit to the whole velocity field. All right, that was just a small multi-scale example. A slightly bigger example, including registration and segmentation. So for this one, I think we used, as I introduced in the background, we used the recently released 10x genomics data set, where they have both xenium data. So again, this is spatial subcellular transcript information, like individual spots. They have immunofluorescence data on top of from serial sections, and also they have from another serial section, they have cesium data, so spatially uniform data. And they, on top of that, they also have H&E. And so we can really work with any type of image registration tools. In this case, we used ST-Align because it's just very flexible in terms of like what type of transformations it allows, both FI, non-rigid, whatever you want. And so we can aggregate, or we can kind of like co-register all of them together, represent them all in the same Giotto object, and then start thinking about what can we do with our framework just to kind of like show a proof of principle about flexibility and usability. So the first thing I thought I'm always interested in, and a lot of people have that question, is like, what kind of like segmentation do you need to use, and how does it change your final results? And so what we did here is we used the original segmentation that was provided by 10x itself, which I believe builds on top of CellPost, which is one of these segmentation algorithms to identify cell boundaries. And now we also use Bezier, which uses these the information, but also on top of that uses transcript location to identify or fine-tune the individual cell boundaries. So here on the left, you see the final results from the original data, and this is Bezier data. So we can jointly cluster them together because we can keep all these different segmentation results all in the same Giotto object. So it's easy to put them all in the same UMAP to them on into, or the same kind of like nearest neighbor network, cluster them all together. So there are no really difficulties in understanding what is happening. So what do we see, and does it really matter? So one thing we saw, for example, in this 10x genomics data set, and maybe I forgot to say this is also a breast cancer data set. So we saw a 100% increase, so a twofold increase when we looked at the Bezier segmentations in stromal and cancer-associated fibroblasts. And so does it matter? I think the answer should be definitely yes, because a lot of people are looking into these fibroblasts right now because they are believed to be essential for the survival of some of these resistant cancer clones after treatment. So just to illustrate you that it might be important that you just analyze multiple segmentation tools or to understand like what are you working with and what do you want to do. And so we make it very easy to do this. And on top of that, you can use this to have more quantitative output about what does it exactly mean in terms of like where the transcripts enrich, what transcripts am I missing. Going a little bit on the sidetrack here, just because I think it's kind of like an interesting exercise in the same direction, using a different technology, actually. I'm showing here imaging mass cytometry. So imaging mass cytometry is intensity-based, right? So you get, it's protein-based, you get an intensity measure at some point. So it's not individual transcripts, but you can assign, you also have polygons, like you see here on the right. You can assign these intensities to these polygons and then cluster them in the same way as you do here. And then this is a zoomed in view. So you can see that you can kind of go through the same pipeline and identify cell types based on the expression of these proteins, which oftentimes is in a lower dimension because you only have like 15 or 20 or 25 of these protein markers. But nonetheless, it's kind of interesting and it's being used all over the world, including for some very large data sets, including the breast cancer space. So one thing we did, using our framework, it's very easy for us to manipulate these polygons in all sort of ways and keep multiple polygons all together in the same Jota object and compare them with each other. So we did a very simple task where we shrink all the polygons by 25%. So they're all slightly smaller, as you can see here. And then we did a similar type of clustering. With the idea is, does it really change the final annotation, or do they more or less stay the same in the same major clusters that they were before? Also, one of the reasons why we kind of like do this shrinking is these technologies, they suffer a little bit from bleeding, where neighboring cells, when they touch each other, for example, what you see here, that kind of like, since its intensity values is not binary for one or the other cells, so you get kind of bleeding from your neighboring cells into the other ones, which is very problematic sometimes to work with. All right, so what are the results? So when we look at this, we see that at least more than 25% of all the cells, they kind of change the majority cluster. Is that bad or is that a problem? We don't really know yet. The only thing I can tell you, if you see, so the ones that do switch clusters, where are they located? And so these are the ones in red. So they're located all over this specific field of view. And does that matter? I think, I wish I could give you a very definitive answer. The only thing I can say is that some of these like data sets that use IMC, but also other technologies, not specific to IMC, it's in many of these technologies, by the way, they like to do niche clustering, where they cluster niches, so cell composition for each individual cell in your neighboring cells, and they assign this to clinical information. And then they say like this specific niche is associated with increased survival or decreased survival. Now, if you look at like how many of these, I think if you look at all the niches between the first and the second one, probably all the composition of almost all niches have changed. So if you assign niches to some kind of like probability of increase or decrease survival or anything, or regression or progression, you might want to be a little bit careful about how exactly you do this. And that's really the only thing I'm saying. This is, again, I'm not trying to make a point. I'm just trying to say, at least our platform allows you to do this kind of like comparison and see how robust certain analyses are and what you can do to potentially improve it in the future.
Ruben, can I just interrupt you real quick, or get a clarification here? Because this, I think, is maybe you're not making a point, but I think you're talking about something very important. So can you quickly summarize? So what was the change that was made for these, like the original k-means clustering? How was the segmentation determined for that one? And then what did you do to modify the segmentation? What was the step that I missed that, like the shrinking? Is this really just shrinking it, or?
Yeah, so we do a very simple shrinking. So you go from, this is one cell, for example, maybe I should have explained this better. And then we simply shrink it in a very simple, maybe simplistic manner of thinking about, let's get rid of the edge effects, which have been shown to be a problem these type of technologies. So all the cells are, so we have the same number of cells in one and the other one. The other one is just a slightly different in terms of like area size. And then we kind of like use the same input data to identify the cells and cluster of cells. And then you just see, do they map? And you do see that many of them map to each other, which is a little bit what you expect. But the fact that even if it's 25% that do not go back to the majority cluster, and also that they are like all over the place, it's maybe a little bit worrisome, or something that we at least should be very careful about when we do this type of big scale analysis and associate it with clinical data.
Right. And then and what was the data type that you used here? So what kind of data modality were you looking at? Is this a
so this is the assay is imaging mass cytometry.
Okay.
So it uses proteins associated with the metal ion, so you get some kind of like a
right. Well, this is I think this is a fantastic example of, I think, you know, the power of having a tool like Jotto in place to actually make these comparisons quickly. I'd be curious, you know, if you get similar results, if you were to use a different type of assay as the input for an analysis like that, you know, maybe some transcriptome-based assay, or another, you know, maybe a microscopy-based assay. That would be really interesting to see, actually, if that changes. But great, thank you for sharing this.
Yeah, and this is, I think we might see this. So I definitely don't want to single out IMC, imaging mass cytometry. I think it's not specific to that technology. It might be more widespread and something that we really need to think about, because it all goes back to the segmentation and how much bias is there potentially in segmentation. But without a framework where you can have multiple segmentations and shrink them and enlarge them and compare them directly with each other, it's very hard to do this. Like most people, they have an issue already, like doing one type of segmentation, and then doing the whole downstream analysis, like that's already complicated. So we're trying to simplify that and make that a little bit easier in the future.
Cool, thank you.
Going back now to the 10x co-registered data set with the Xenium data, the IF data, the Visium data. I'm going to show you very simple again. These are all like very simple type of analysis, but made very easy to do by others using our framework, where we're going to compare modalities or technologies. So very simple, what they provide is, though, they have their Xenium data that's with specific transcripts that you can identify here on the right. For example, this is the CD20 transcripts. These are the ERBB2 transcripts, or HER2 transcripts. On the left, you see their corresponding IF data, so immunofluorescence data, on an adjacent slide or a nearby slice from the tissue. And as you can see, that the agreement, I think, is pretty good, although the R-square, or the Pearson R, doesn't really show it for this one, because there is some kind of like, is it background or is it something else? We don't really know. It's a little bit better for this specific one. Similarly, since we can use these co-registered data, you can also compare it between the Xenium and the Visium. And now you have a little bit more to work with. And here is one gene, FASN, on the Xenium slide. This is the Visium slide. So on the Xenium slide, we aggregated in the same type of spot-based manner as for Visium. So you can make a very accurate description between the two. So it's not apples against pears, but it's kind of like apples versus apples, similar apples. And so one thing we did is we also looked at, let's do it for all the genes, right, that are available in the Xenium and have overlap with Visium. So Visium is kind of like agnostic to specific genes. So it picks up whatever it finds using a poly, or is it FAPE? I might be wrong here. But Xenium, we have about 300 genes, if I remember correctly, maybe a little bit more, maybe a little bit less. And so these are like, what you see here is correlation values between all the gene pairs between Xenium and Visium. And you see there's a pretty good correlation for most genes, but it kind of like drops all the way down to some of them have no correlation at all. And that was interesting. We're like, oh, is that because like they are maybe different type of genes? Is it difference in type of like how they are regulated? But it really seems, if you just like plot the expression levels of these genes in Xenium and Visium, you can clearly see that there's a very strong correlation between correlation and Visium with the expression levels itself. So genes with high expression levels in Xenium and Visium, they typically also like have a very good correlation with each other. While if you kind of like drop down, you obviously are looking now at some kind of like noise levels. So it's going to be very hard to get that really good correlation levels. So that's yet another example. A last example is kind of like joint clustering. That is something that has been done previously, but we have also, that's also now easy to do within Giotto in all sorts of ways, where on the same slide, we're not going to use this specific data set, but we're just going to use a data set from 10x where they have both protein and RNA on the same exact slice, where you can use the RNA matrix, the protein matrix, and you can kind of like do these joint or bench reduction and do some kind of like weighted nearest neighbor or any type of network integration analysis to use both modalities to cluster your cells together. So in this case, this is a tonsil data where you can use only RNA or protein to cluster your data, but then within Giotto, our Giotto suite, you can combine these networks. And one example that we implemented is the weighted nearest network, but you can do all sort of different type of integration of networks analysis, and then use that multimodal information to view it. Just to show that we can work with this like adjacent slices, but can also be the modalities can also be in the same tissue slice or can be at different tissue slices. So everything is really possible. All right. Okay, let's go to interoperability. So interoperability is really a priority for us. I think we also like to use other tools for certain analysis, and so we made, or I think we have something that works at least between Giotto Spatial Experiment, Giotto Seurat, and also Giotto, and right now it's the N data. Hopefully in the future we might expand this a little bit more to, for example, the Spatial Feature Experiment, which is something that is kind of like similar-ish to Giotto, as well as the kind of the spatial data that was recently developed by the people from the VUES, which I think is going to replace some of these N data objects. Just as an example, just one example here, like I think Bento was developed using an N data framework. And it's kind of interesting because one of the few tools that really tries to look at subcellular localization, tries to identify patterns, subcellular localization patterns within this type of spatial subcellular data. And so we have made it very possible, and we made it easy to kind of like go to this Bento tool and kind of like identify if certain transcripts are enriched in nuclear, cytoplasmic cell edge, and so on. All right. Okay. So this is really why I like to work in R and why I don't want to switch to Python. Although some people think they're trying to convince me to go back to Python. I think that our spatial ecosystem is really unprecedented. It's really huge. It's been there for like decades, and it's really amazing. It's super powerful. And so Giotto is kind of like built on top of some of the data classes in Terra, although we can kind of give it our own flavor, or like really big type of own flavor, and try to make it even better. In addition to that, we also have built-in converters between the Terra classes itself, and as well as stars as SF, the simple features, and all of them together, I think more or less represent 95% of the whole R spatial ecosystem. So we have direct access to all of these geospatial data packages, which have a lot of really great tools that are really amazing to work with. And I think if anything, I would like to say, if I have seen further than others, it's by standing on the shoulder of giants. So we really don't want to reinvent the wheel, but we want to use and leverage that type of work and kind of like provide it to the community, provide it to people working with spatial omics data. So just as an example, this is just in the Terra package alone. There are so many different spatial specific statistics, metrics, tools, algorithms available that you can immediately use or almost immediately use within the Giotto suite framework. And I'm going to highlight something that we did in less than 2 weeks, and I think the results look really great. It's kind of like using some of that spatial interpolation that has been used all the time, where you try to impute missing information between points for which you have spatial information. So I'm going to give you that kind of like strategy that we used using this kind of like background. So in Giotto suite, you can just analyze your typical, this is the mouse brain Visium data set. This is one specific gene. So you can do your typical analysis and visualize that specific gene. And that's great. But as I showed you before, it's very easy to have a matching H&E image, use any of your segmentation tools, and also like identify individual nuclei in that H&E matching H&E. image. So that's number one. So you can use that as another spatial unit. So one spatial unit of your Visium would be simply the individual spots from your Visium array. Another one could be the identified nucleus, and you can like expand a little bit so you have an individual cell. So the other thing is the R spatial ecosystem that I just talked to you about, where you can do spatial interpolation. And in a very simple way, kriging is one of them, where you try to identify missing information between sources of information that you have. So that's exactly what you have with Visium, right? With Visium, you have these individual spots with information, but then there's actually a missing spot, and then there's another spot. And there's a very coarse solution. Kriging allows you to impute that data, but also allows you to enhance its resolution in a very simple and effective manner. So if you combine these two within in the Shelter Suite framework, you can go from what I showed you before, so this typical Visium data set, to something that looks like this, where you now at the single cell level, a single cell nucleus plus some like extract, you can visualize these genes at the single cell level rather than the kind of like the single spot level. You can do that for all genes, at least all genes that have some spatial variance. And if you do that for all genes, you can go from something like this that you've probably seen many times, where you can see a very nice layer structure in your Visium spots, to something that looks like this, where now all of a sudden you go from, and it's much better to see this in the zoomed or the subsets, where you have the Visium all the way down. Now this is like individual cells. So in a very simple way, we can super enhance our Visium data sets using this type of technology based off on everything that we've implemented in the Shelter Suite framework. And this allows us to build on top of that and make it even a lot better, including some of the morphology data and so on. All right. So that was the interoperability part. I think we still have a few minutes, right? So the interactivity, we use that really to create what I call a reactive environment. And I think that's really important. Is you're doing your kind of like interactive analysis. You're trying things multiple out. You're clustering. You're seeing clusters, but then sometimes you just see something, you cannot really get it out with some unbiased clustering. So now you can launch your Shiny application. You can draw these polygons in whatever shape you want. You can have multiple, and you do that, and that is immediately integrated into your Chilto object. So you can use it in the spatial transfer analysis without having to switch between one application to the other one. So it's all integrated within the same kind of like Shelter Suite interactive exploratory data analysis. In the same way, you can also do 3D in silico slicing, where if you have a 3D data set and you want to kind of like understand like what is a 2D representation, if you slice it this way or that way, in whatever way, you can do that as well as keep that information and use that in your downstream analysis. And then I think last but not least is kind of like the issue that is like, it's great to have this kind of framework, but having all this additional information and all putting it together in one big framework makes it also like a problem for scalability. And I think that is an issue that comes back more and more all the time. And spatial data are often very big data. So here I'm showing Chilto, what we have done for spatial genomics, for nanostring, for SeqScope. So it all works. And we have implemented some of the functionality from the bioconductor. So we do use bioconductor quite a lot, and we try to be, as I said before, interoperable as possible, including some of the delayed area and future apply. But it's still kind of like can be very problematic. So on top of that, we implement our own spatial chunking approach so that we can very efficiently even like analyze very large data on a local machine. But ideally, you might want to use your supercomputer, or you want to use the cloud, where we have also like installed and made Chilto available on the Terra cloud from the Pro at the moment. And as a last example, I just want to show you something that kind of like goes hand in hand, is that Chilto also has very flexible ways of tiling data in all different sorts of forms, whatever you can think of, and all like you can change these parameters if you want. And I'm showing this for a StereoSeq data because it's really something where it's very valuable. So StereoSeq is Visium, but it's kind of like Visium in steroids, where you have 500 nanometers arrays with a lot of information in these nanometers. But you do need to find some way of like aggregating these individual transcripts, right? So we can easily introduce hexagons like hex bins, and this is like a 200 scale, at 50 scale, and you can pick the one that you like the best and you can keep working. So for this one, we did that. I think this is on the 50 scale. You can get a nice patterns where you see exactly like all those different anatomical regions that you would expect in a full mouse embryo. But I want to show you a little bit more what you can do next with this like flexible tiling approach, where you say like, maybe I'm very interested in this specific region. So you see this very typical, this is a brain region, this brain and this multi-layered, but now you want to use a more resolved or higher resolution hexagon. So you can easily like add that to your object and kind of like go a little bit deeper, where you see multiple layers all of a sudden at once. You can also like create a pseudovisium, as I showed you before, where you can use a different type of tiling strategy. It's not really tiling per se, but where you can now make a pseudovisium from your original stereo-seq data. So where you can assign that, and then that gives you the opportunity to use that information for benchmarking, where you have a ground truth data set. You can also benchmark your, in our case, we have Spatial WLS implemented, but you can use whatever deconvolution approach that you want, and you can benchmark it immediately with your stereo-seq data, giving you an idea like how well do some of these deconvolution approaches work. So all of that, these are all like different examples where like tiling and making it very easy to go from one spatial unit to another one and keeping them all together is very useful. All right, and I think last but not least, I think the real issue is if you really want to make it so that it's very flexible and everybody can use it, I think we need to think about data sets and infrastructure that are really scalable, but also that are democratic and green and are accessible to everyone, and that you can start right away without doing anything. And this is not really part of Filter, but something we're working on, where we think about more using specific databases that can support some of these matrix operations and spatial operations that we're doing, including maybe sequencing in the future, where there is a very flexible backend where you can use all type of databases that can feed right into Filter Suite. And I think that is probably more like kind of like the future. I think that the field is going, or maybe should be going, where the backend that you use can be really whatever you want, but it's very flexible, in that the overarching organization, in our case, Filter Suite, kind of like remains the same, so that people don't necessarily have to learn a different type of like structure. Right, I think a few slides just to end. So this is work done by many people in my lab. The lead developer is George, but he has a really fantastic team of other people helping him implementing all these different tools and examples. We do this together with GCUN at Mount Sinai, where George's counterpart is Jocelyn, where she helps us implementing a lot of new tools and ideas. And they also have a bunch of new people. We also get support, generous support from many funding agencies. We are about to submit, I wish I could kind of show you that was already submitted, but unfortunately it's not. But we're going to put it on bioRxiv really soon, maybe tonight, maybe tomorrow. And last but not least, please take a look, let us know if something is not working, or if something is not working what you want. We are very interested in thinking about building a more like an open source governance structure, where it's not just us deciding what to do, but more like there's also other people contributing. And we have more and more people actually doing this, so we're going into the right direction. Last but not least, maybe this is a good idea to also think about building something, a unified spatial metadata framework for MEMS. So rather than basically saying like everybody needs to have exactly that spatial data structure to work off, I think it's probably a good idea to have more like a metadata framework, where as long as we know this is kind of the metadata we need, you can use whatever backend and whatever technology that is available in the future to operate on your data in the future. And that's it. All right, thank you.
Right. Thank you so much for the presentation, Ruben. This really a lot has happened since we last spoke. Very exciting to see that.